Alrighty, hi guys. Welcome to my video, uh, DNA replication part 2, PCR and amplified products. So the part 1 video talks about DNA replication in the cell and how that works. This one talks about the second half of the story that we're working with this week, which is PCR. So if you haven't seen that other video, I recommend going to watch it because PCR makes a heck of a lot more sense if you understand how things already work in the cell. So if you need help understanding how the cell works, make sure to check out part 1. Okay, so for starters, let's go over what PCR stands for. PCR stands for polymerase chain reaction. Now go over what each of those mean. Polymerase means that we're using a polymerase enzyme in this process. If you remember uh, from DNA replication in the cell, the polymerase enzymes are the enzymes that read the DNA, the original DNA strand, and build the new DNA strands to complement it. And the specific enzyme that does that in the cell is called DNA polymerase 3. So we're actually going to be using DNA polymerase 3 again here. So the polymerase stands for DNA polymerase 3. And then a chain reaction, if you've got any background in chemistry, you'll know that a chain reaction is a chemical reaction that once started automatically continues to happen over and over again until stopped. So PCR means we're going to be using polymerase to do some sort of reaction that'll happen over and over again until we stop it. So it's a process that uses DNA polymerase enzymes to cause a chain reaction. And in this case, the chain reaction that we're doing is linking DNA nucleotides together in a long chain to form a new strand. So we're going to use polymerase to build a new strand, essentially. Now, how is that different from the cell? The difference here is we're doing DNA replication in a test tube instead of inside a cell. So we've eliminated the cell from the story here. We're just trying to copy DNA inside of a test tube. Now, this allows us to collect a sample of DNA from the environment or wherever, and we'll talk later in future weeks how you can do that. But you can collect a sample of DNA, and then if I want to photocopy just a specific sequence from that sample, I can make like a million copies of that particular DNA sequence. You can think of PCR kind of like a Xerox machine for DNA. The idea is I'm trying to find a specific like if I'm interested in a particular gene on the DNA, a lot of the processes that we're going to talk about in future weeks require lots of copies of DNA to make them work. So we use PCR to make lots of copies. So essentially, you're just going to Xerox that one gene and make a bajillion copies of it so that we can work with it. Now, what do we use PCR for? Because in the readings, they kind of talk a little bit about how it works, but they don't really talk about why it's important. And I have a lot of students that ask me, like, so why do we care? It turns out PCR is actually one of the foundational processes that we use in molecular biology. I'm a molecular biologist, and I use this basically every day. PCR revolutionized uh, the biological industry. Let me show you some of its applications here. For one, next week, we're going to talk about forensic analysis and how you can use DNA to track criminals um, and things like that. And so we can use PCR to do that. And again, we'll talk about how we can do that next week. But a lot of the processes that we will teach you about next week involve large quantities of a copy of a particular region of the DNA or of the chromosome. So you would need to collect a sample of criminal's DNA, make lots of photocopies of that particular region on that DNA, and then that can help you see who the individual is. Paternity testing works on the same idea. If you're trying to figure out who your parents are, or for example, if you send in your DNA to one of those websites like ancestry.com or 23andMe, to get your DNA tested to find out your genealogy or whatever, they have to do PCR in order to find, or they do PCR on certain segments of your genome to help them figure out who you're related to. And paternity testing can work the same way. Like if you're trying to figure out who the father is of a particular child, um, then you can do that through PCR as a preliminary step. We, of course, use it to study genes. Scientists that are trying to figure out how genes work can make lots of copies of that particular gene, and then using techniques that we'll talk about in week 10, can utilize that to study how the gene functions. For the same reason, we can use it in genetic engineering, which we'll talk about in week 10 and week 12, um, where we can make copies of a gene and then move it into another organism to give them a trait that they didn't have before, which is really exciting. And so PCR can be super helpful there because you can make lots of copies of the DNA to help put them another into another organism. We'll learn in week 9 how you can use PCR to do medical disease screening. So we can figure out if you've got some sort of genetic disease like cystic fibrosis or sickle cell disease by using PCR. And PCR is a prerequisite to sequencing, and it's part of DNA sequencing. Um, and so you can use DNA sequencing, you can use PCR in general to help diagnose if you've got a particular genetic disease. And we use it to help detect viral infections. Most recently, the COVID-19 pandemic has been raging throughout the world. And they actually use PCR in COVID-19 testing. So most of us have probably had it where they took that swab and they stuck it up your nose, and it was painful and miserable, right? And and then they carry that off and you get these test results back. Well, what they're doing is they do PCR on the sample that they took in the little swab to see if they can amplify any DNA from the COVID-19 virus. And if they succeed, that means the virus was there. And so you test positive. And if they don't succeed, that means the virus wasn't there. And so it was negative. So that's kind of a more recent application there for PCR. And there are a lot more examples, but hopefully this will give you an idea that PCR is actually foundational to all that we know, and all that we do here in the biology field. So now that we've talked about what we use it for, let's actually get into the meat of how it works. So I'm going to switch over to my simulator here. Alrighty, so here's my simulator. So just to kind of orient you to what we're looking at here, right here, we've got a double-stranded piece of DNA. So these two blue lines represent the two strands of a DNA helix. So just bear with me. I did this all in a spreadsheet because spreadsheets are basically all I know how to do. So bear with me here. So there's our double-stranded DNA, and you'll notice that the 5' and 3' ends are labeled of each of the strands, and that'll become important here in a sec, because we learned about that last week. And then you'll notice over here on the right-hand side, I've got a thermometer, which is also going to become important because we use heat a lot in the PCR process. So keep an eye on that thermometer as well. Okay, so there are 3 steps. So let's say I'm trying to make a photocopy of this middle segment right here. Like, let's say this middle segment of DNA is some gene that I want to study or something like that. So I want to make a lot of photocopies of that particular segment. So we're going to do PCR to make copies of that. Now, in DNA replication in the cell, you learn there are a bunch of different enzymes for that we use to make it happen. Like you have to have helicase to unwind the DNA, and primase that that builds the primers, and then DNA polymerase 3 comes in and reads the DNA and builds a new strand. And then you have to have DNA polymerase 1 come in and cut out the thing, and you've got topoisomerase to make sure that it doesn't get kinked, right? There are all these different enzymes, and to for PCR to make sense, you need to understand why we do what we do, and this is a very critical point. In the cell builds all of those enzymes. Well, if I'm doing this in a test tube, if I'm trying to do DNA replication in a test tube, that means there's no cell to build those proteins. So any proteins I use, I have to buy. And there are actually companies that will sell you enzymes. You hop on to their website and you can buy these enzymes, and they'll send you a little tube like this that's got like a little droplet at the bottom, and ta-da, there's your enzyme. The problem is enzymes are very expensive. They can, for a droplet, they can range from like $70 to like $300 per enzyme, depending on what you're looking for, which enzyme you're using. So they can be extraordinarily expensive. And believe it or not, we do try to cut costs in science. So we want to try and eliminate as many enzymes as possible from the DNA replication process while still accomplishing the same goal. So a lot of the things we do here, keep that in mind. We're trying to do the same thing they do in the cell, but eliminate as many enzymes as I possibly can while still being able to accomplish it. So if you remember, the first step in DNA replication in the cell is helicase, has to bind to the DNA and unwind it, right? You get a replication fork and whatnot. Well, we want to try and eliminate enzymes. So we're going to try and eliminate helicase. Well, but we still have to separate the DNA strands. So it turns out that when you heat up anything enough, it melts, right? DNA is the same way. Turns out that if you heat it up to 95 degrees Celsius, the DNA strands simply float apart, because we learned that the backbones of DNA are held together by strong covalent bonds, but across the middle, they're only held together by weak hydrogen bonds. So if you heat it up to 95 degrees Celsius, the weak hydrogen bonds break, but the covalent bonds are stronger, so they don't. So the strands will simply drift apart. So let's show you that here. So we're going to heat this up. In the first step, we heat it up to 95 degrees Celsius. If you look over here at my thermometer on the right, we heated it up to 95 degrees Celsius. And notice my DNA strands separated from each other. So now I've got two single-stranded bits of DNA. So the strands just simply drifted apart from each other. So that's the first step in PCR, and it's called denaturing. Now, this is a term that we use for separating the DNA strands, and the way you could think of it is when sorry guys, I'm a little out of it today. The natural state of DNA is to be double-stranded, right? We learned about that. So if you are denaturing something, that means you're undoing its natural state. So denaturing means we are undoing its double-stranded state. Hence, we're heating it up to separate the strands into their two pieces. So the denaturing step is the first one because we have to separate the strands. Okay. Now, so we've eliminated helicase while accomplishing the same goal. Now, your cell can't do this. It needs helicase because if we heat you up to 95 degrees Celsius, that's close to boiling water. And so that's kind of lethal, right? If you have a fever of 95 degrees Celsius, you're dead, right? So your body has to use enzymes. But in the test tube, it's not alive, right? So we can heat it up to 95 degrees Celsius and break the rules there. Okay, so if you remember then, after we've separated the DNA strands, the next step is you need to lay down a primer because DNA polymerase 3 doesn't know how to start a strand brand new. It only knows how to make a pre-existing strand longer. So you need a primer. Now in the cell, we had an enzyme called primase that built that primer. However, remember, we're trying to cut as many corners as possible. So we want to try and eliminate primase if possible. However, we still need primers. So how can we do that? Well, it turns out that there are scientists that are a lot smarter than me that run companies that make primers. So I can actually hop online and tell the company, here's the sequence of the primer that I want, and order it. And it costs about like $5. Of course, it's probably getting more expensive these days because everything's getting more expensive these days, but it costs about $5. And they manufacture my primers, send it to me in a tube, and I can dump it into my reaction. And ta-da, I've got primers. So I eliminate primase because primase would cost like $70, whereas the primers, just buying pre-made primers, costs only $5. So we saved money there by simply ordering already constructed primers and then just dumping them into our tube with the DNA that we're trying to copy. So we've got these primers and they're floating around in this tube here. The problem is we need them to bind to the DNA, right? I need them to come in here and stick to the DNA on either side of my sequence that I'm trying to copy here. However, we're still at 95 degrees Celsius over here. And the whole point of heating it up to 95 degrees Celsius was DNA strands float apart from each other. So while we're at 95 degrees Celsius, those primers are not going to be able to stick to the DNA. So we got to cool things back down again. Oops. Deleted that on accident. Okay, there we go. So we've got to cool things back down again. So that's the next step. So the next step, we're going to cool it down to 55 degrees approximately. It depends on the primers that you're going to use, but for the purposes of this class, it's about 55 degrees, usually in the mid 50s. So this second step, we cool it down to 55 degrees, and that's cool enough that my primers that I dumped into my tube can come in here and stick to the strand. Now notice there's two different primers involved in the reaction. You always use two primers, one on either side of the sequence that you're trying to amplify. So this second step here is called the annealing step. So the word anneal is the term that we use when DNA strands stick together. So the annealing step is when you cool it down to 55 degrees Celsius so that the primers that are just floating around in the tube can come and stick to the spots that they're complementary to. So that's what these two little blue bars, these two little, the shorter blue bars represent. These are our primers right here. Okay. Now, at this point, I'm going to pause here because you're probably wondering why the primers are sitting where they are. It turns out that when you design primers, there are a couple of rules that you have to follow. And if they don't follow all of the rules, the pair of primers isn't going to work and your PCR is going to fail. Okay. So you need to know these three rules because they're going to help you on your homework. Wink, wink, wink. Okay. So here are the three rules to designing a functional pair of primers. Rule number one, the primers must bind to opposite strands. Primers must bind to opposite strands. What that means is of my two primers, because remember, we're always using two primers, one of the primers has to stick to the top strand, and one of the primers has to stick to the bottom strand somewhere. If both of my primers stick to the top strand, like if I put a primer there and a primer here, it's not going to work. If both of my primers stick to the bottom strand, like if I put a primer here and a primer here, it's not going to work. I have to have one primer stick to the top strand, and I have to have one primer stick to the bottom strand. That's the first rule. Okay. Okay, let's clear those out here. The second rule is that the primers must be on either side of the sequence you want to copy. So in this case, we're trying to copy this middle piece. So I need one primer on the left side of the piece I'm trying to copy and one primer on the right-hand side of the piece I'm trying to copy. And I am not writing well today. Okay. Primers must bracket the sequence to be copied. Another way of saying that is your primers can't stick in the same spot. For example, if I design my primers so that one of them bound right here and the other one bound right here, I'm passing rule one. I've got them binding on opposite strands. I got one sticking to the top strand and one sticking to the bottom strand. However, they're both sticking to the same spot in the DNA. They're not on either side of the sequence that I'm trying to copy. And in fact, in this particular case, if I tried to use these two primers, you'll notice that since this one down here, the bottom one sticks to the bottom strand at this spot, and the top one sticks to the top strand at this spot, those two primers are actually complementary to each other. So what would happen is I would dump them in the tube, the primers would stick to each other and essentially eliminate themselves from the equation, so nothing would happen. I officially wouldn't have primers in the tube. So we don't want that, right? So the primers must bind on opposite sides of the sequence you're trying to copy, or if you don't have a specific sequence you're trying to copy, just make sure they're not sticking in the exact same spot, okay? That's rule number 2. Rule number 3 is probably one of the trickier ones to keep track of. The 3' end, or the 3' ends of the primers must point inward. Or in other words, they must point towards each other. For example, if I laid down a primer right here and a primer right here, they're passing rules 1 and 2, right? I've got a primer on the bottom strand and I've got a primer sticking to the top strand. And they pass rule 2 because they aren't sticking to the same spot. They're sitting on either side of the sequence I'm trying to copy. However, notice here, if we look at my 5' to 3' orientation, the 5' end of this bottom primer here is on the right, and the 3' end is over here, and the 5' end of the top primer is on the left, and the 3' end is on the right. So if we drew an arrow here, the 3' end of this primer is pointing off to the right. The 3' end of this primer is pointing off to the left. Notice they're pointing outward. They're not pointing towards each other. So this pair of primers won't work because it violates rule 3. Now, let's look at the pair of primers that my simulator put down. And let's see if they will work for us. These are the 3 rules. Let's see if the primers will pass all 3 rules. Do they stick to opposite strands? Yes, I've got one on the top strand and one on the bottom strand. Do the primers bracket the sequence to be copied? Yes, neither of them are sticking in the same spot. I've got one on the left and one on the right, so that passes. Do the 3' ends of the primers point inward towards each other? Well, let's look. This primer, its 5' end is over here. This primer, its 5' end is over here. So this one, if we're going 5 to 3, it's going this way. And this one, if we're going 5 to 3, is going this way. So they are pointing inward, which means we pass rule 3. So this pair of primers will work and actually successfully make a product, which is what we call the photocopied piece of DNA. So this pair of primers would successfully make a product. If a pair of primers breaks any one of the three rules, it will not make a product. It will fail. Okay. So that'll help you with the last problem on your collaboration assignment. Keep these three rules in mind. If a pair of primers breaks any of the three rules, it'll fail and won't make a product. Okay. So let's clear these rules out here to make room to get on to the next step. So we've cooled things down. We've laid down our primers. Now the next step is to build the new strands. And we learned in the cell, this is done by DNA polymerase 3 coming in to bind to the DNA here at the primers. Now, of course, this is PCR, so we are trying to eliminate as many enzymes as possible to make it cheaper. The downside is scientists have yet to find a way to replace DNA polymerase 3. We aren't that good yet. We still need DNA polymerase 3, hence polymerase chain reactions. So DNA polymerase 3 is actually the only enzyme that we use in PCR. We've found little tips and tricks to get rid of all the other ones. So DNA polymerase 3 will come in here and we're going to we still have to buy it, so it's expensive, but we buy the enzyme and we dump it into the tube. So now DNA polymerase 3 is in there. Now we need it to build the new strands. Now, we're not just using any DNA polymerase 3. And here's the trick here. What made PCR possible was actually this particular discovery, and it's a really cool one. Turns out that, remember how we talked about if I heat you up to 95 degrees Celsius, you die? Well, part of the reason for that is most proteins fall apart at such high temperatures. And human ones, in particular, will fall apart. The proteins will unfold and they'll cease to work. And unlike DNA, when you cool it back down again, proteins are so complicated that they have a really hard time folding back up again properly. And so once you denature a protein, it's basically broken and done for. So we don't need the polymerase to actually work until step 3. But step 1, we have to heat the DNA up to 95 degrees Celsius, which means if I'm going to use a DNA polymerase 3, it's got to be able to not fall apart at 95 degrees Celsius. And that's the big innovation with PCR. It turns out that some scientists went to Yellowstone, and there's a particular hot pool there at Yellowstone. And you've probably noticed several of these hot pools. This water, they've got these hot springs at Yellowstone, and it's steaming, right? And there's steam coming off of them. These lakes are basically close to boiling in temperature. However, if you look at a picture of these Yellowstone lakes, they've got like an orange, they're colors inside the lakes. And a lot of that is due to bacteria living in the lake. So there are bacteria actually living in this near boiling water. And some scientists put it together. They were like, wait a second, bacteria need DNA polymerase 3 to duplicate their DNA. So if there are bacteria surviving in these near boiling lakes, that must mean they've got a DNA polymerase 3 that's built in such a way that it doesn't fall apart at 95 degrees Celsius. So they collected a bacteria from the lake called Thermus aquaticus. Thermus aquaticus. And so that's the name of the bacteria. And they took that bacteria and they stole its DNA polymerase 3. So we call that enzyme, because that's the one that we're going to use, we call it Taq polymerase. We're going to take away that here, and we're going to call it TAQ. It's tacky, I know, right? Because that stands for a DNA polymerase that came from Thermus aquaticus. So the TAQ just stands for which bacteria we stole it from. So Taq polymerase is the exact same thing as DNA polymerase 3. The only difference is it doesn't fall apart when you heat it up to 95 degrees Celsius. It's heat resistant. But it works the exact same way. You still need primers. It's still going to build in the 5' to 3' direction. All polymerases build in the 5' to 3' direction. Remember that. That is a hard and fast rule. There are a lot of caveats in science. This isn't one of them. You always, always, always build in the 5' to 3' direction, whether you're Taq polymerase or DNA polymerase 3. Okay? So we're going to use Taq polymerase, which we also dumped into the tube, and it's going to come in here and it's going to bind to the primers and it's going to build 5' to 3' on both of these strands. So this step here is called extension. Okay. I did spell it wrong. That's awkward. Okay, extension. So notice here, it hopped on and it built my new strands. Now, one thing you're going to notice here is I didn't copy anything before the primer. That's because we have to build in the 5' to 3' direction. And so anything that's before the 5' end of the primer isn't getting copied. Now, if we were in the cell, we would solve that problem with Okazaki fragments, right? I would come and lay down another primer somewhere over here, and then we'd build 5' to 3' and fill in that gap. But in PCR, we don't need to do Okazaki fragments because remember, I'm not trying to copy the entire chromosome. The cell, when it's doing DNA replication in the cell, it needs to copy the entire chromosome because it wants to try and pass an entire copy of its genome to the new baby cell. In PCR, our objective isn't to copy the whole chromosome, it's trying to photocopy one gene. You might think of it like DNA replication in the cell is copying an entire book. And here in PCR, I'm trying to make 1 million photocopies of one page of said book. So I don't care that we didn't copy anything before the primer. As long as my 2 primers are sitting on either side of the sequence that I want copied, we're going to be fine. And I'll show you that here in a sec. So, and it'll actually become important here in a sec that we didn't copy anything before the primers. So be aware of that. It's important that we didn't copy anything before the primers. Okay. So now I went from one copy of one double-stranded piece of DNA to 2 double-stranded pieces of DNA. I've doubled it. Now, the next step in DNA replication in the cell is you got to come in and cut out your primers with DNA polymerase 1. Well, here we did another shortcut. Why do you have to have DNA polymerase 1 come in and cut out the primers in the cell? Well, it's because primase builds the primers out of RNA, and we can't have RNA sitting in our double-stranded DNA strand permanently, right? So we have to remove the RNA and replace it with DNA. Well, in PCR, I'm not restricted by the God-given mandate that primase built with RNA, right? Because primase builds with RNA because God said so. I'm sure there's a reason for it. I don't know why, right? God in his wisdom did so. Well, I'm not having primase build my primers. I'm having a biochemist in Ohio build my primers, right? So he's not restricted by that mandate. So I can just have him build my primers out of DNA instead of RNA. So then I don't need DNA polymerase 1 to come in and cut out my primers and replace them. So I've saved money there too. So in the cell, The primers are made of RNA. In PCR, our primers are made of DNA. We could use RNA primers. There's nothing wrong with using RNA primers. However, this saves us money because now I don't have to cut said RNA primers back out again. So in PCR, we choose to use DNA primers simply as a convenience measure. So keep that in mind. These primers are made of DNA, not RNA in PCR. So we're done. That's one round of PCR. We did all three steps. Now, remember, we're trying to make 1,000,000 photocopies of this. Oh, I forgot to point out. DNA, notice over here on the thermometer, we heated it back up to around 72 degrees for the extension step. And that's because that's the temperature that Taq polymerase works at best. So we warmed it back up again so that Taq polymerase would be happy. So you always change the temperature for each and every step. Now, what's really nifty about this is we only leave it at each step for like a maximum of a minute, usually less, actually. So you warm it up to 95 degrees for a minute, and then you drop it down to 55 degrees for a minute, and then you warm it back up to 72 degrees for a minute. So it only takes 3 minutes or less to do one round of PCR, which is fantastic. Now, we talked about earlier, before I got sidetracked, we talked about how you have to have like 1,000,000 copies of this. And right now, I've only got two, which means we're going to have to do this again. The convenient thing is, I've still got more primers floating around. I've still got a bunch of bases floating around. We call them DNTPs, which stands for deoxyribo, meaning DNA, nucleotide, which is just the individual bases. And then the TP stands for triphosphates. So what this means, if you remember back to week 2, and sorry, I know this is a bit of a sidetrack, but I forgot to cover it earlier. The structure of a DNA nucleotide was we had a sugar, we have a base, and we've got a phosphate, right? Those are the three pieces of a DNA nucleotide. Well, it turns out that this is the structure of a DNA nucleotide in, or when it's actually built into a DNA strand. But when it's floating around just as a free building block that hasn't been built into a DNA strand, it doesn't actually look like this. It actually has three phosphates right in a row out here. And that's important for reasons you don't need to know. Basically, these phosphates act as a battery pack that contributes the energy that allows polymerase to stick each base onto the end of the growing strand. So you don't need to know that, but people have been curious what DNTP stands for. Well, it's deoxyribo, that stands for DNA. The N stands for nucleotide, and then the TP stands for triphosphate. So it's a DNA nucleotide with three phosphates on it. That's what a DNTP is. So DNTPs are basically just the free-floating nucleotides in there. So we've thrown in our primers, we've thrown in our DNTPs, our nucleotides, so that polymerase has something to build with. So all we have to do this again is start the cycle back over again. We heat it back up to 95 degrees Celsius, and that's going to separate our DNA strands. Now, just to keep track of my simulator here, these two long strands are the original strands, and then this strand right here, this one right here came from up here, and this one right here came from right here, okay? So, or sorry, from right here, my bad. Okay, so just keep track of that here. So I separated those strands, and notice two of them are shorter, because remember, we didn't build anything before the primer. So now we're going to do step 2. which was the annealing step. So now our primers are going to come in and stick down. Then we do step 3, we do extension. Now notice, again, we didn't copy anything before the primer, right? Here or here, and we didn't copy it over here or over here. So notice when I do this again, now we've gone up to 4. When I do this again and heat it up, now I've got strands of several different lengths. I've got the ones that are the original length right up here. Sorry, I should draw. Let's switch over to drawing here. Okay, so we got these ones up here that are the original length. Then we've got this weird one here, and we got this weird one here. Actually, we've got these 4 right here. These 4 right here are kind of shorter. They've been clipped on one end or the other. And then I've got these 2 right here that have been clipped on both ends. That's going to become really important here when we finish this step out, okay? So let's finish this out. So we're going to have our primers bind, and then we're going to extend. Now notice, on these ones down here, we polymerase bound to the primer and then ran until it ran out of strand. And it bound to the primer and then ran until it was out of strand, right? So now we've got these segments right here. We have all of our bizarre ones, our strands of random length up here, but now we've got these 2 new ones that I marked here in gold that are the length of the leftmost primer plus the piece in the middle that we were trying to copy plus the rightmost primer. So left primer plus right primer plus everything in between. This is what we call the amplified product, which is the other thing that we discussed in our or in the title of my video here. This is called your amplified product. And it's always the length of the left primer plus the right primer plus everything in between. So, for example, let's say up here, because you'll see something similar to this on your homework. If I broke this up with a ruler, and let's say this over here is 20 base pairs long, and this here is 10 base pairs long, and this here is, I don't know, 30 base pairs long. It's not the scale, right? I'm just making this up off the top of my head. Okay, and this is 10, and then this over here, sure, we'll call it 20. So if it's asking me, if the question asks me, what is the length of my amplified product? Now, remember, I was trying to copy this 30 base pair segment here in the middle. However, my amplified product is not 30 base pairs. And it's not the 90 base pairs that the whole thing is. Instead, it's from the left edge of the leftmost primer to the right edge of the rightmost primer and everything in between. So it would be 10 plus 30 plus 10. So my amplified product in this case would be 50 base pairs long. That's how you'd figure out the length of your amplified product. It's always the left primer plus the right primer plus everything in between. So don't forget, the primers are included in the length of your amplified product. Because of this, we always copy from the left edge of the left primer to the right edge of the right primer, and you wind up with these golden pieces that are your amplified products. Now, why do we call them an amplified product? Because you'll notice we made lots of other pieces that are of different lengths. Why do I call these golden ones here my amplified product? The reason for that, let me show you, if we go one more round, let me show you what it looks like if I do a 4th round. Okay, So we heat it back up again, separated all the strands. We land the primers there. Now notice, I've still got the same number of these weird strands up here, right? There's still the same number of them. However, I've got more gold strands down here that are the length of the amplified product. So every single time you do another round of PCR, we're going to keep the same handful of these weird strands up here at the top, and then I'm going to keep getting more and more and more of these gold strands exponentially. After I've done, say, 30 rounds of PCR, I'm going to have like, what is that, 8 weird strands and a bajillion amplified product size strands. So really, we can ignore the 8 weird ones, and all you wind up with are the amplified product size ones. So that's why we call it that, is your PCR product is the amplified product. It's going to be that length right there, the golden pieces. Now, we've talked about how we double the number of strands that you make every single time you do PCR. So if I was asking you how many copies of DNA you'll have after a certain number of rounds of PCR, you could simply do your original starting number times 2 times 2 times 2 times 2 times 2, right? But we've got a little formula here to make that easier. Here's the formula for figuring out number of copies after PCR. Okay, here's the formula. X times, that little dot means times, 2 to the power of n, where X is the number of strands or the number of copies you started with. And n is the number of rounds of PCR. Now, this you'll notice this formula is the shortcut to what I was telling you earlier. Because remember, we're doubling it every single time you do a round of PCR. Well, the way you could show that is the number of strands you started with times 2 times 2 times 2 times, right? Or what that is times 2 to the number of times you did it, right? The to the power of here is how many times did you multiply it by 2? So that's essentially what this is. We're taking the number of strands and we're multiplying it by 2 this many times. That's what the formula means. So let me show you an example here. My computer program says that after I did 4 rounds of PCR, I've got 16 strands of DNA. Let's see if the formula tells me that. So in my particular example, we started with 1 double-stranded piece of DNA. So X is 1. So I'll do my little formula down here. Okay. So in my particular case, it's 1 times 2 to the power of, I did 4 rounds of PCR times 2 to the 4th. Let's pull up a calculator and see what that is. Come on. You can tell I'm really tech savvy, right? Okay, here we go. So we're doing 1 times, I'm going to put this in parentheses here, 2 to the power of 4 is 16. So that formula can tell you. Now, just to give you an idea of how crazy this can be, when I extract a piece of DNA from like a tissue sample or from a saliva sample from a crime scene or something, it's not just going to have one double helix, right? There's going to be lots of DNA in there. So let's say I start out with like 30 copies of it, right? So let's say my X is, or sorry, yeah, my X is 30. So X is 30. And let's say my N is 35 rounds, because typically, at least in the lab when I do it, we do about 35 rounds of PCR. So if I were to take 30 copies of my DNA strand and do 35 rounds of PCR on it, I'd come out with 30 times 2 to the power of 35. That's a lot of copies. 34 billion strands of D or 34 billion copies of the gene. That's enough to work with, right? So PCR is extremely effective. And it takes getting to higher rounds to see. But if you keep doubling it, you wind up with a heck of a lot of DNA by the time you're done, right? So that's the point of doing PCR. Um, so yeah. So if you've got any, hopefully this video helped. I'm sorry that I rambled a little bit, but if you've got any questions for me, please feel free to reach out on Inscribe or to reach out to your teachers. And we're all happy to help. So thank you for watching.